Hello everyone. So I'm back with another video after a really long time, and I hope I am able to explain the concepts well. So I would like to start with a very basics of biology, which is a central dogma of biology. Okay, so the central dogma itself means it's the central basis or the main rule of biology. Okay, which means that this is the basic funda of biology. यही होता है, ठीक है. It's like a rule कि ये follow होगा ही होगा. So ये किसने दिया था? By it was given by Francis Crick. Okay, so what did Francis Crick say? He said that once information had gone from DNA into the protein, it could not get out of the protein and go back into the genetic code. So मतलब इसका मतलब क्या हुआ कि हमारा जो जेनेटिक मटेरियल है दैट इज डीएनए, ओके, विच वाज प्रूव्ड बाय एवरी मैक्लेयर्ड मैकार्थी एंड फ्रेडरिक ग्रिफिथ एंड फ्रेडरिक मिशर. There were many people who contributed in the improving that DNA is the genetic material, and also to find out where the genetic material is located, etc., the model of the structure. Okay, what are the advantages, disadvantages? Is RNA a potent genetic material? Okay, so if you want a video on that, please let me know in the comments the topics you would like me to take up, and if I can, I'll surely take it up. So okay, so he is saying कि हमारा genetic material है DNA. So from the DNA, which proteins are made will be the proteins will be made, and once the proteins are made, it cannot be कि I think we should move forward, and then you'll be able to understand it better. I'll come back to this statement again. So, have you ever thought, see, our muscles are made up of proteins, okay? The transport of material from cell to cell happens via membrane proteins, right? There are membrane proteins, there are transporters, okay? Most of them are made up of proteins. There are hormones which are protein hormones, okay? Like oxytocin, it's a protein hormone. Then there are enzymes, okay? Enzymes, protein enzymes. Then there are, yeah, our phenotype is regulated by protein, right? We have a skin color, okay, melanin. Then we have eye color, okay. These have pigments which are proteins in nature, right? So protein, surface proteins of the cells, okay, all these play an important role. So all this forms a phenotype, and your phenotype is proteins, right? So we studied that skin color and eye color, ये सब तो genetically acquired होता है. मतलब when you get the genetic information, you gain these phenotypic characteristics, right? So how is it? Because of proteins, but also because of genetics, right? मतलब genetic information and proteins are somehow interrelated. So how is that interrelated? It means that whatever information our genes are carrying can be transferred to the proteins, or the genetic information is telling us that which proteins are to be formed. Okay, so this chain of the transfer of information from DNA to protein has one intermediate RNA. So DNA, mRNA, DNA forms mRNA, mRNA forms polypeptides, that are proteins. So this is the central dogma of biology. So probably now you will be able to understand what Francis Crick was trying to say, right? He was saying that information goes from genetic material to proteins, but not from proteins to genetic material back. Okay, next. So both these statements are correct. Traits are controlled by proteins, and the synthesis of these proteins is controlled genetically. Does it mean that our genes, DNA, which are nucleic acids, are controlling the proteins? Okay, so how do these so this means that our genes are controlling the proteins. So how do nucleic acids control amino acids? So by controlling we mean that genes are responsible for the synthesis of protein chains. Okay, so the clear explanation is given by the central dogma, which is DNA by transcription will form RNA, which by translation will form protein. Now see, DNA is a nucleic acid, RNA is a nucleic acid. From nucleic acid we are going to amino acid. So basically it's like a language is changing. First it was just transcription, the language was same, the script changed. Okay, it was nucleic acid, but first it was DNA, then it became RNA. Now we are changing the language from nucleic acid to amino acids. That is why it is translation. Okay, you can remember it like that. Then DNA will form a RNA strand, which is called as a messenger RNA, which is the mRNA, and it gives a message to the ribosome, and the ribosome is involved in the synthesis of proteins. So there are three processes. First, DNA to DNA synthesis. Second, DNA to RNA synthesis, which is transcription. DNA to DNA is called as DNA replication. Then we have RNA to protein synthesis, which is called as translation. So this is a very brief explanation of DNA replication. So firstly, polymerase is enzyme polymerize the DNA and RNA. So if it is forming a DNA, we call it DNA polymerase. If it is synthesizing an RNA, we call it RNA polymerase. If it uses a it also nomenclature also depends upon which template it is using, मतलब वो क्या पढ़ के बना रहा है. So if it is reading a DNA template and making a DNA molecule, it is DNA dependent DNA polymerase. If it is reading DNA and synthesizing RNA, it is DNA dependent RNA polymerase. Okay, so these polymerases are able to construct only in the 5' to 3' direction. I hope you are clear that the DNA has two poles. Okay, DNA has a polarity because the last sugar at if possible, I'll make a video on what is how is the polarity formed in DNA. Okay, so 5' end होता है जिसमें phosphate होता है, 3' end होता है जिसमें hydroxyl end होता है. You can remember it as 4 for 5 and 4 for phosphate. Okay, so 5 phosphate ऐसा याद रख सकते हो. So क्या होता है, the parent strand having 5' to 3' direction makes a complementary daughter strand to itself, that is 3' to 5', which cannot be made by polymerases, right? See, अभी क्या होता है कि if you can see, this is the 5', this is 3'. तो इसका कॉम्प्लीमेंट्री कैसे बनेगा 3' to 5'? ये उल्टा है, राइट? It's not possible. Now this is a DNA polymerase enzyme. Suppose this green colored ring which you see, suppose it is the DNA polymerase enzyme. So ये polymerase यहाँ पे attach हुआ, ठीक है? And यहाँ पे था ये पहले, and it is moving in this direction. तो ये आगे आगे जाएगा, बट इसको अगर हमको इस स्ट्रैंड को अगर हमें कंस्ट्रक्ट करना है इन 5' to 3' डायरेक्शन, इसके कॉम्प्लीमेंट्री स्ट्रैंड को, तो वील हैव टू सिंथेसाइज इट दिस वे. इधर 5' आएगा, इधर 3' आएगा, राइट? तो ये ऐसे आएगा opposite to the direction of the fork. It is not possible because the enzyme is here, right? So there is an enzyme, there are different types of DNA polymerases, and there is something called as beta clamp. Okay, so the beta clamp is attached. Okay, the RNA primase. Okay, RNA primase. See, also polymerases require a primer, which means वो suddenly एक base add नहीं कर सकता. There should be pre-existing few nucleotides which are already present. Okay, it's called as a primer. तो वो प्राइमर से आगे कंस्ट्रक्ट करेगा. तो यहाँ पे उल्टा कैसे होता है ना, पॉलीमरेज जब यहाँ था वो एक प्राइमर लगाएगा इधर, ठीक है, सॉरी एक प्राइमर लगाएगा इधर एंड उल्टा सिंथेसाइज होगा, फिर आगे एक प्राइमर लगेगा और उल्टा सिंथेसाइज होगा अवे फ्रॉम द डायरेक्शन ऑफ द फोक, एंड ये टुकड़े टुकड़े में होता रहेगा. इट इज कॉल्ड एज लैगिंग स्ट्रैंड. पर एज दिस स्ट्रैंड इफ यू सी, ये ऑलरेडी ऐसे 3' to 5' है. इसका कॉम्प्लीमेंट्री बनाना है तो इधर से 5' to 3' बनाना पड़ेगा, और पॉलीमरेज भी उसी डायरेक्शन में जा रहा है. तो ये जाते जाते एक एक बेस ऐड करते जाएगा, तो भी ये कंटिन्यूसली सिंथेसाइज होगा. तो जो कंटिन्यूसली सिंथेसाइज होता है वो लीडिंग स्ट्रैंड है, जो टुकड़ों में सिंथेसाइज होता है दैट इज द लैगिंग स्ट्रैंड. एंड दीज पीसेस इन विच द लैगिंग स्ट्रैंड इज मेड इज कॉल्ड एज द ओकाजाकी फ्रैगमेंट्स, and it is named after the scientist who discovered it. Okay, so yeah, this is the replication fork. Okay, this is because it forms a y-shaped structure, it's called as the fork. So this is a very common question asked, that in which direction the leading strand is complementary to the leading strand is synthesized, uh, and in which direction the lagging strand is synthesized, and what is the polarity, what is the template, what is the coding strand. Okay, I'll discuss polarity and uh, DNA, how DNA gets its polarity, and how to what is coding strand, non-coding strand, template, non-template. I'll make a video on that also if required. The next strand, this is the difference between the lagging strand and the leading strand. I'm sure that if you go through it once, you will be able to understand it. It's not a very difficult thing. Whatever I have explained before is the same thing. Note, in both the strands, the daughter DNA molecules have one parental strand and one new strand. Hence, we can say that half parental is conserved in the half parental information is conserved in the daughter cells, right? Daughter DNA. So it is semi-conservative. Okay, and there are other modes of application as well, dispersive and conservative, which were proven to be wrong. This is transcription, and I am not going into much detail of transcription. I'm just giving a brief idea about it. If you need any detailed information on one particular topic of transcription, please let me know in the comments. So we have this is a DNA. Blue color जो है, that's your DNA, and it has bases. And this red color is mRNA, which is now being synthesized, okay, from the DNA. Now DNA is being read and RNA is being made. So this becomes DNA dependent RNA polymerase. Okay. So these are the yellow are the bases. So RNA polymerase is the enzyme. Okay, you can see this gray color in the background is the RNA polymerase, which binds to the DNA, and the bases are added complementary. One difference you should note is RNA always has uracil instead of thymidine. So U will be added opposite to A, wherever T is supposed to be there, वहाँ पे U आएगा, right? So A का complementary is T, so instead of T in RNA you will have U. Then we have when the RNA is completely formed. Okay, now transcription does not occur throughout DNA. DNA has some repeating sequences, some junk DNA sequences, non-coding sequences, जिसका transcription नहीं होता है. They don't code for any protein, so there is no transcription of that area. So this mRNA, the RNA synthesis happens of very few particular segments of DNA, and these are called as genes. Okay, these are the functional segments of DNA. And when this mRNA is formed, it has exons and it has introns, which are again intervening sequences which are not required. Okay, so it is like exons. When you join these exons, you get a nice mature mRNA which is coding for a peptide nicely. ये introns जो है वो intervening sequences, and these are not required. So these are then removed by splicing. Splicing is done by catalytic RNA. Okay, and these are called as spliceosomes. So these introns will be removed, and you will get this orange colored complete exon containing mRNA. So these are the steps. First, there is initiation. The RNA polymerase will bind to a sequence called as the promoter. Okay, and where there is elongation, please go through these texts. I have written it in very simple language, and it is very basic. I am very sure that you will understand. And if you don't understand, and then you have any doubts, you can always ask me in the comment box. Then we have translation. So RNA to polypeptide formation is translation. So there is a ribosome 50-30 in prokaryotes, 60-40 in eukaryotes, and the mRNA which was formed in the previous step will come and bind to the ribosome. There are three sites APE. Have I mentioned it? Okay, I have not mentioned it. So there are three sites APE. Okay, amino exit site. Okay, the peptidyl site and the charged amino acid tRNA site. So amino acid tRNA site. So the amino acid are added to the tRNA and it is called as a charged tRNA, which will come and sit in the A site. At the P site, whatever amino acid it is carrying, उसका पहले वाले previous amino acid से peptide bond बनेगा, hence called peptidyl site, and the exit site where the empty tRNA will leave the ribosome, and continuous polypeptide chain will be formed, which will be released, and then there will be it will go to the Golgi complex, and there will be protein sorting, and there will be folding, and with the help of chaperones the protein will get its shape and then it will become a functional protein. This is just the primary structure of the protein. Okay, it undergoes folding to form a secondary structure, tertiary and quaternary structure, and a final functional protein is formed. That's all for today, and thank you so much for watching this video. Please let me know your queries and requests in the comment box.